I'd like to welcome everyone to the BEEc Speaker Series. This is an international webinar series hosted by the Center for Bee Ecology, Evolution, and Conservation at York University. So a brief bit about BEEc. As I mentioned, we are the Center for Bee Ecology, Evolution, and Conservation. If you'd like to learn more about our mission statement, our outreach activities, and other research activities, please feel free to visit our website, yorku.ca/bees, or reach out to me, the coordinator, at bc@yorku.ca. Very briefly, BEEc is an initiative that strives to advance research in the field of honeybee ecology, evolution, and conservation. And the mission of BEEc is to foster interdisciplinary, innovative, collaborative, and cutting-edge research. This research is used for the advancement of knowledge and implementation of policy changes to help sustain pollinators globally. And our goal is to apply our collaborative efforts to the development of policies and environmental management for the long-term sustainability of bees and the vital ecosystem service they provide. If you'd like to hear more about the activities that BEEc hosts or get our monthly newsletter, please subscribe to our BEEc Global Listserv by sending an email to listserv@yorku.ca with the following command in the body of the email: subscribe bees, and then your first name and your last name. If you are a York University researcher or collaborator, please visit our website and see if you would like to become a member of BEEc by filling out our associate application, or even learning about the benefits of what it would be to become a BEEc associate. Once a month, on the second or third Friday of the month, we host our BEEc Speaker Series at 11 a.m. Eastern. These are Zoom webinars, so you'll have to register for each one of them on our website. And we have an exciting roster of speakers for our fall semester, including our speaker today as Dr. Adam Dolezal, Dr. Laura Figueroa in October, and then our November and December speakers as well. You can register for all of these webinars, or at least the October webinar, on our website, and the November and December registrations will be coming out soon. Additionally, we're working on getting together our roster for the winter semester, so I'll also be sending out messages on our listserv about when we have those ready to be registered for as well. So for the logistics of the speaker series talk today, you all can see we are using a Zoom webinar platform, so only the panelists can turn on their cameras and microphones. If you'd like to ask a question during the talk, please use the Q&A box to pose questions to the speaker, and Dr. Zayed will pose the questions back to the speaker at the end of the presentation. Also, you can pop messages into the chat box, but please use this sparingly just for general comments. And if you're having any trouble with the webinar, please send a private message in the chat to me, the host, Laura Newburn, or you can email bc@yorku.ca. This presentation will be recorded and posted later to our YouTube channel. Additionally, on our YouTube channel, we also host videos from previous webinar series, like our BEST series, our previous Associate Speaker Series, as well as videos from our yearly conference, BEEcon. Now, before we get started today, we would like to recognize that many Indigenous nations have longstanding relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territories of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples, and we acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you would like to know more about the history of land that you come from or where you are joining us from today, this website, native-land.ca, is a fantastic resource. It was initially a Canadian initiative, but it has worldwide coverage now. And now I would like to welcome Dr. Amro Zayed, a professor at York University, who will introduce our guest speaker and who will lead the Q&A session after.
All right, thank you so much, Laura, for that lovely introduction. Welcome, everybody. We're really excited to launch this speaker series for this term, and it's a great pleasure to introduce Professor Adam Dolezal. Adam is an associate professor and Centennial Bee Research Scholar at the University of Illinois at Urbana-Champaign. He's in the Department of Entomology. He completed his PhD in Arizona State University, followed by a postdoctoral work at the Iowa State University with Dr. Amy Toth. His laboratory focuses on how pollinators respond to different stressors that they encounter in the environment. And these studies have been really crucial for understanding how viral and nutritional stressors influence bee health, especially in Midwestern systems. So please join me in welcoming Dr. Adam Dolezal. Thank you, Adam.
Thank you. Yeah, thanks again for inviting me to speak. It's really exciting to get to come and to talk to such a cool group. Like Amro said, I am here at the University of Illinois, and this title is a little bit of a joke in my lab where we have this all these experiments that we do, and I always have to tell all the new people in my lab that everything sounds simple, but nothing is ever easy because of how complex some of these systems can be. So again, I first want to start by thanking everybody in my lab. I've been really lucky. I started my lab in 2017, and since then it's grown kind of a lot, and I've gotten to have quite a nice group of people, some of whom I'll get to highlight today. And I, without my knowledge on our website, the picture that will greet you is this one on the right, which was a April Fool's joke that my lab played on me, where I came to work on a day where we had our own seminar, and my entire lab was dressed as me. It's not clear to you guys who don't see me every day, but I wear the exact same clothes every day, which they all are wearing. So it's been a kind of a fun journey the last 7 years here at the University of Illinois. And just, I wish this picture wasn't so blurry, but to highlight the big shoes to fill and the kind of bee research to live up to. When I got my lab here in 2017, it came with a lot of stuff, and in that stuff was this pile of racks, as you can see, highlighting Amro's rack right here. So the physical lab space I inhabit was the lab space that Amro worked in when he was a postdoc here. And we still have a bunch of his stuff, and we get to use it every day, which is fun. So some of the things that I've been interested in since I started my scientific career were how different types of insects respond to or how they respond with different phenotypes to different environmental stimuli. And like Amber mentioned, I did my PhD at Arizona State University, and I actually focused primarily on harvester ants there and studying how the social environment and the reproductive physiology interacted. But at the same time, I got some experience working on honeybees and kind of got my first taste of that. And so when I was looking for postdoc positions and kind of to think about where I wanted to go with my career, it was a time when it became increasingly important to study pollinators, but because of my kind of base interest in this, they really were a great and ideal system to work on, because we know that pollinators, honeybees, wild bees, butterflies, they're exposed to a lot of different types of environmental stresses and different types of environments, spanning from differences in their nutritional landscape, exposure to agrochemicals, exposure to pathogens, and then for honeybees, the social environment that they create and kind of have within their own natural biology. I probably don't have to remind people of some of these slides in this audience, but as we all know, honeybee losses have been continued to be above acceptable thresholds for beekeepers, with beekeepers reporting losses that are considered to be economically difficult to sustain, and so the costs of beekeeping have become much greater. At the same time, we have lots of evidence for widespread declines in native bee populations that are probably experiencing a lot of the same environmental stressors, but are often much more difficult to study. And so we've probably all seen this figure adapted from AAAS and originally coming from the Schoolson paper from 2015, highlighting that bee declines in this paper, quoting, saying, bee declines are driven by a combined stress from parasites, pesticides, and lack of flowers. And he kind of highlights here some specific pesticide risks, but I think they can be kind of generalizable. And what I really like to highlight here is that, this paper is now a decade old, highlighting that interactions between these factors are what really likely drive a lot of these declines. And yet it still remains very challenging, and we still have the relatively limited ability to study how these things interact. And so that's really been the focus of my lab for the last 6 to 7 years. And really the postdoctoral work I even did before that was finding a way to look at these factors both by themselves and also try to study how they interact together to give us these more complicated phenotypic responses. But it's really hard, and so it requires a lot of different types of studies and different types of experiments to start to tease apart what factors are important and how they interact with each other. And so I want to tell a couple stories about projects that we've done in my lab that look at these alone or in conjunction. And I'll talk a little bit about this more when we get to the limited floral resources, but we know that bees are exposed to pesticides and nutritional deprivation very heterogeneously depending on where they live. They're in agricultural areas or more natural areas. That can be very highly variable. But for honeybees, almost ubiquitously, we have pressure from parasites and pathogens, primarily from viruses that are transmitted by varroa mites. And no matter where you live in much of the world now, that pressure is going to be there. And in my lab, we primarily study pathogens through use of a tool that we and others have started to develop of using Israeli acute paralysis virus as kind of a virus model, because it exhibits some really nice pathogenic pathologies in bees that are very easy to observe, very easy to measure, and they occur very rapidly. So this is from a methods paper from one of my students, and we see that, and this will become important as I show data later, when you infect a bee with Israeli acute paralysis virus, they either die or they don't within 3 days. If they don't die after 3 days, they're probably going to survive the infection, and if they are going to die, they will have died before that. And so this just really makes them a useful model for understanding how response to a pathogen is modulated by different other things. Because unlike things like Nosema cerani or even deformed wing virus, those pathogens can take sometimes weeks to manifest really observable symptoms. So it makes it a lot more challenging. And so we've done a lot with this IAPV model. And I'm going to tell a couple stories a little bit about that. And so the way that I've started to really conceptualize this is this idea of a disease triangle, which people in disease ecology should be pretty familiar with. But when I started working on bees, this was somewhat new to me. And it's this idea that to have disease, we have to have these three different points of the disease triangle. And so, we have this idea that you have to have a susceptible host and a pathogen and an environment, and those kind of three things need to be aligned to present diseases. And so that's kind of the realm that we've thought about it. We've also started to study one of the implicit aspects of the disease triangle, sometimes conceptualized as a disease pyramid, which is to add time as a factor. So like I said, with this disease model, what we've been able to be really successful with it is by developing our own methods and using some developed by other researchers to really purify and propagate Israeli acute paralysis virus very easily without having to use a really good cell line, which is often used in other insect virology systems, by just extracting white-eyed pupae or slightly pink-eyed pupae from normal colonies. These bees, if they don't have a mite on them, usually have very low virus titers. We can then inject them with IAPV or deformed wing virus, propagate virus particles that way, extract them, and then use them in tons of different kinds of experiments. And so it's given us a lot of flexibility in studying how virus infection responds to these other factors. Like I said, we can then take this virus, propagate it in the pupae, purify it, and then we can infect adult bees in tons of different scenarios and by a couple different methods. We can feed it to them in sugar solution, either as a group or individually. You can make them regurgitate and then manually feed them. Or you can inject it into them in very small levels to mimic a varroa-mediated infection. And so again, that gives us a lot of flexibility. As I mentioned, we've been kind of working on this as a model because of its rapid infection timeline, where, like I just mentioned, but you didn't see the slides of, we see infection really manifesting within 36 to 48 hours, and it's pretty much done by 72 hours. So our replication can be really high. So one of the interesting things to me when I started working on this is that people have been talking about honeybee viruses as a major problem for bees and for bee health for, again, over a decade. But there are actually still a lot of major gaps in understanding really basic disease ecology aspects of the infections. And the reason I kind of guided me in some of these was I was working with a mathematician here at the University of Illinois on a project that I unfortunately don't have time to talk about modeling bee disease transmission and parasite transmission. And she would ask me all these questions like, when a bee gets infected, when is the peak point for virus titers? When are they most infectious? If they die, what are their virus levels compared to if they survive? And then, what happens to a bee who survives the infection? And the answer we had was that we didn't really know those. We didn't know the answers to a lot of those questions, but we could find out. And so just to kind of give a little bit of background in that, we did these experiments with my postdoc Alex Payne and a PhD student Vincent Perugo, looking at the titration or titering virus levels and seeing when we see peak infection. Again, this used to be kind of harder to explain before COVID, but now we know that identifying when a host is having its peak viral load and is therefore most infectious is really valuable for predicting how infection moves. And so one of the things that we see here, if you just look at bees that are day one, handling control, they've been injected with what we call HKV, heat-killed virus, or if they've been injected with IPV. And we see that within 1 day, IPV titers, this is a log scale, are huge. Their virus titers go way up. They're very, very high after 1 day. However, if you look at that over time, we'll see that they really peak in that 24-hour period, and then they decline very rapidly. And what we see is that if a bee does not die of an infection within 24 to 48 hours, she appears to completely clear the IAPV infection. However, let's see. This is sorry, I'm having some Zoom problems. You can see this kind of play out here, that if bees are dying of this infection, their infection occurs and causes effects within like 3 days, and then it really flatlines after that. Although we've investigated this a little bit more with some more nuance to it. But the moral of the story is that they survive, they don't appear to die of infections. What about deformed wing virus? Certainly we know more about that. However, in most of our surveys of the literature, there haven't really been a lot of explorations to show that really baseline data on what happens when bees get infected with DWV as adults. Usually they're infected as pupae, they exhibit these deformed wing symptoms, but we know that you can infect a bee as an adult. And so we did the same kind of comparison, and what we see is a very different response. So on the first day, virus titers are actually not that elevated after you inject them with deformed wing virus. It takes some time, about 2 to 3 days, for them to reach peak virus titer, but then it stays high for, again, the duration of these experiments. Interestingly, we also don't see any mortality in these bees until like day 6 or so. So the moral of the story here is that here are these 2 different bee viruses that often co-occur, but their infection dynamics and their pathology for adult bees is extremely different. They're reaching peak titers at different times. They're causing mortality at different times. And so the way that we predict or kind of model pathogen transmission and pathogen effects is going to be very different for these different viruses. And I don't have time to go through it today, and it's somewhat new and a little bit convoluted, but we've been doing some more experiments now looking at co-infection of these viruses, because in these experiments, we're often looking at them only kind of in a vacuum, but in the real world, we know that DWV and IAPV almost always co-occur. And so, again, I don't have time to talk about all these different projects, but we set the groundwork with some of these virus pathology questions, but we also then did a bunch of different projects on seeing how these responses can be changed by exposure to other environmental changes. So, for example, we did a project several years ago showing that the type of pollen that you feed bees causes a significant effect on how their survivorship is affected, both in kind of model pollens, which we see on the left, but on the right, these are pollens that are pulled directly from our real world landscapes. And so again, I won't go through these in much detail, but you can see that if bees consume no pollen, their survivorship is much less. And depending on what pollen they do consume, we see differences in how much better they survive. And this again seems really intuitive, that if they have better nutrition, they'll survive better. But there is some complexity to it. We also found that some pesticide exposures like flupyridiferone, which is a relatively non-toxic insecticide, does synergize a little bit with infection and changes those virus infection dynamics. So again, the moral of the story here is that lots of things can change these responses. We also were interested in some more complex phenotypes. So Israeli acute paralysis virus, as you can tell from its name, is hallmarked by paralysis symptoms. So a bee gets infected at high enough levels, she exhibits shaking, paralysis, and eventually death. However, the sublethal infection pathology are much less well understood. And so we had a project that we did a couple years ago that explored this in greater depth. And the main thing I want to highlight here is modulation of how IAPV infected bees are treated by the members of other colonies. So normally when a honeybee colony is out there, it has guard bees that stop and prevent foreign bees from entering the colony. And that's what we see in this figure here, proportion of bees accepted by the guards. And we can see that bees who are just handling control or bees who were fed double-stranded RNA to stimulate their immune system, they get rejected quite often. But bees who are infected with IAPV are actually accepted into the colony at almost double the rate. And so we tested this in a bunch of different ways, but my favorite is this very field experiment where you use this little box that we got from Cassie Vernier, who did some nice work on nestmate recognition for her PhD. And we can set up here and see this green marked bee being interrogated and stopped by guard bees. And we can just follow her for the duration of the experiment and simply rank, did she get accepted into the colony or thrown out? And we can see this one is getting attacked. It's pretty unambiguous. Other bees just walk right in. But we see that when bees are infected with IAPV, they're much more likely to be allowed into foreign colonies, which we think has important implications on how IAPV is spread and will be really beneficial for the virus. Another way, though, that the virus might be changing its host biology for its own benefit is through affecting developmental time. So this was work done by my master's student, Lincoln Taylor, who's now working with Sam Ramsey in Colorado. But this is, I'll kind of walk through this somewhat confusing figure. So we started doing this work trying to infect bees as larvae with Israeli acute paralysis virus, because previous work had shown that IAPV's tissue tropism is such that it's highly localized in the hypopharyngeal glands, where royal jelly is produced by the nurse bees. And so we hypothesized that it may be being transmitted to the larvae. And so we did some experiments trying to infect them, but nothing ever happened. And we were at first worried we weren't doing it right. We had all these concerns. No bees ever died. And so that's what we can see here is that if you start a bee on day 0, so right, it's first day after hatching, we can rear them in the lab as larvae. We're giving them a diet that is contaminated with IAPV at very high levels for the entirety of their development, the entirety of their larval development. So this 5-day window. Here's when the inoculation period ends and this bee becomes a prepupa. It stops eating. About day 8, it normally enters into a white-eyed developmental stage, then becomes a pink eye, kind of purple eye, and then eventually, after between 19 to 22 days, it's an adult bee, right? And so in these experiments, we fed bees either virus, heat inactivated virus, or just a PBS control. And then what this figure is showing is the proportion of individuals who are in this white-eyed stage. And so you can see, until the 8th day, nobody is, right? They're larvae or prepupae. At the 8th day, we start to see no matter what group they're in, they start developing into these wide-eyed pupae. And then if you look at the red line or the black line, kind of our controls, a lot of them are wide-eyed, and then by day 13 or 14, none of them are wide-eyed. They've now become purple or pink or purple or adult bees. However, in the bees who have been treated with IAPV, they just stop developing at the wide-eyed stage. They just totally arrest, and they never develop any further. However, they don't die. We've measured this with gene expression and a couple other different ways. They remain alive for this duration of this time period. However, they never develop any further. When we look at the virus levels in these bees from these different groups, we see some interesting patterns too. If you look at a larva, this is a larva who's been swimming in IAPV contaminated food. She's been eating virus food like her entire life. If you take her body and you dissect her gut out, her body has extremely low virus levels compared to the control in red. There's almost no detectable virus. However, if you look at her gut, unsurprisingly, it's full of IAPV. She's been eating it her entire life. However, when we then let her develop to the wide-eyed stage, we can just cut them in half and look at the head and thorax versus the abdomen. Then it no longer matters. The virus titers are super high throughout the entirety of the pupa. And so we see this weird phenomenon where IAPV is not causing a systemic infection in the larval stage. However, it seems to be hanging out in there and persisting until the pupal stage, where it can reach extremely high levels and then cause developmental arrest. Now, why would developmental arrest at the wide-eyed stage be valuable? Previous work by people like Humberto Boncristiani and Olaf Ruppel have shown that wide-eyed pupal stage is the best time to amplify IAPV particles. It is more successful in that stage than it is at other stages, and it's more successful than other bee viruses at that stage. And so at this point, we're kind of hypothesizing that IAPV is causing this weird pathology where it arrests development in the prime developmental stage for its own replication. And so again, we're seeing that the virus had this very weird phenotype that's different than just causing paralysis. And this is something I'm really excited to keep working on. And again, one of my students is exploring the kind of the mechanisms behind this in greater detail right now. And so the moral of the story from all these is that just alone, when we just look at how different bee viruses affect bees, there's still a ton to learn. We only kind of have scratched the surface with the basic pathologies that occur. We don't really understand the timeline. And so we still have a lot to learn if we want to predict how these viruses affect honeybees and then potentially other bee species that are also infectable with them. And that's just one factor. And so now I want to talk a little bit about this issue of limited floral resources. So this is an issue that is especially prevalent in the agricultural systems that I have lived and worked in, and in fact grew up in. I'm originally from Illinois, where the landscape is really dominated by corn and soybean agriculture. And so when we put beehives or have wild bees in these situations, what are the resources they have available to them, and how is that going to impact their overall health? Like I said, this has become incredibly notable in the area that I've been working in. This is Iowa and Illinois. This is yellow on this map shows acreage of corn. And so as you can see, we're in this extremely high concentration. But this is something that occurs across a lot of the US as well as other parts of the world. Currently, about 40% of the United States is dedicated to agriculture. But when we look at our Midwestern Corn Belt states, it's much higher. In Iowa, it's about 86% of the land is agriculture. In Illinois, we're about 75%. And in the area of Illinois I live in, which is right here, it's on the high end of that. One way to try to conceptualize this is I found it often difficult with people who are from other parts of the United States, other parts of the world, where they have a lot of agriculture, but they also have a lot of other land mixed in, to ask, well, why does Illinois look like this? Why does Iowa look like this? It's because the opportunity cost for conservation and for other land uses is extremely high. So a weird way to conceptualize this is in the United States Department of Agriculture, every year, they make a big list of the average rental price per acre of farmland of different types in every county in the United States. If you take that list and you sort it, in the top 100 counties in the US for farm rental prices, 39 of those counties are in Illinois, 47 of them are in Iowa. And the average per acre rental cost is in Illinois $235 per acre, while at the national average it's $79. And so if you are a landowner, either farming the land or renting it to someone else to farm, the land in this region is amongst the most productive in the world for this type of agriculture. And so that the economics of that have played out, and that's why we have very little else in these landscapes. At the same time, we also have really high honeybee wintering losses and now annual losses across these states, probably for a variety of reasons. But we were really interested in whether these landscapes can even really sustainably support bees at all, and if so, when were the times of really good and really bad opportunities for bees. And so at the same time, these areas have been identified as kind of terrible for wild bee abundance and wild bee diversity. We've done some work on that. I don't have time quite to talk about it, but it just highlights that these areas are under extreme stress, but they're also really a useful kind of landscape model of what I would consider, from a diversity standpoint, a worst case scenario. And so we did this really large project in Iowa. This was started back when I was at Iowa State University, where we looked at how the surrounding landscape around apiaries affected the growth, development, and health of bee colonies. And we did this. This is, again, a map of a real site. Yellow is corn, green is soybean, gray is roads, and this light gray or light green is not those things. And so you can see this landscape is just corn and soybean and the little grass edges around them. And we looked at areas of what we called high cultivation and areas of low cultivation, which I highlight that are low cultivation areas. These are areas that still 50% of the land around an apiary was corn and soybean agriculture, but that's as diverse as it gets. And again, I think that's sometimes hard for people to wrap their mind around if they're from a region that is a little bit less extensively farmed like we have here. Now, interestingly, what we found is that areas of low cultivation actually did slightly worse than areas of high cultivation. But the thing I really want to point out across these is that no matter where bees were kept in our landscape, they reached this peak in July and then saw a significant decline in their mass between August and October. Now, to some extent, this is normal, but the magnitude of the decline was really extensive. And it's because we thought that the landscapes we were in, after the crops are done, there are no resources. This is a weed-free environment. It's a conservation-free environment, so there's very little else in the landscape. To test this, we then looked at what would happen if you replace some of that land with what used to be there before. So Iowa, before European settlement, was about 80% tallgrass prairie. Currently, it's less than 0.1% tallgrass prairie. So it's a land use in a kind of ecosystem that has pretty much been eradicated from the landscape, but can occur and does occur. Now, and when we put honeybee colonies in these with access to these natural landscapes, we saw a really significant response. So here we can see that this is the change in weight from their maximum. And so we can see around this time in mid-July, colonies are at their maximum weight. They're starting to lose weight. And then we randomly moved half of our colonies to an area where they had access to a prairie. The other half did not. Those who did not move continued to decline in mass, while those who have been moved to a prairie had this big boost. We see a similar boost, slightly delayed, with fat stores of individual bees. And so there's opportunities for bees to get better resources in these landscapes. However, they're at this point extremely rare. One of the things I want to point out about this study too is that while we gave bees access to prairies, there is nowhere in these areas where they're insulated from agriculture. Prairies are not very big. And this is a picture of our real field site. You can see it's beehives next to a big prairie. But across the street over here is a gigantic soybean field and a gigantic cornfield. They're completely surrounded on all sides by conventional agriculture. So they're not insulated from some of the other kind of inputs that go into that agriculture. And so what's been really interesting to me then is to ask, well, how do we find ways to support bees, both honeybees and wild bees in these super dominated ag landscapes? And one argument is, well, maybe we shouldn't worry about it, and we should just accept that those areas are completely dominated by agriculture, and that allows us to have conservation elsewhere. And that's a totally valid argument. But at the same time, that's where I live. And so I would like it if there were some areas that were not completely agriculturally dominated, and that we could create some kind of conservation for wildlife in these areas. The reality, though, is that currently we have like 22 million acres of crops, and it's very unlikely that those are going to be turned into conservation land on a broad scale. And so I was really interested in trying to study kind of policies or land management strategies that would be consistent with our modern agricultural system, but still have real benefits and actually be economically viable. And so the main thing that we've looked at this is through this system called STRIPS, or prairie strips. It's kind of a weird backronym of science-based trials of row crops integrated with prairie strips. But we can see a picture of this right here. So here we have a conventional field, and then incorporated into it are these strips of basically tallgrass prairie restorations. Here's another picture showing what they can look like. Another couple pictures in soybean on the right and corn on the left. And what's really interesting about these, to me, is that they were created and promoted by some people at Iowa State, like Lisa Schulte Moore, not for pollinators, and not even really for wildlife, but initially for erosion control and for nutrient runoff. But at the same time, as you can see from just looking at it, this is a great potential habitat for bees and other pollinators. However, there's been a huge concern about it. This concept called an ecological trap. So when you have an area like this, where you create really great habitat, but it's surrounded by something that could be detrimental, like pesticide use in an agricultural setting, you actually have the potential for an actual conservation detriment, where a habitat like this is going to be highly attractive to bees. They're going to come in, use the food resources, use the nesting resources, and then a month down the road, a large-scale pesticide application is going to be done to these crops, killing all of those bees. And that's been a huge policy question in the implementation of these strips: is, are they actually even valuable, or are they going to be detrimental? So we did this big project with collaborators at Iowa State that had a lot of different features to it, and I'll only talk about a few of them. And first was just measuring pesticide exposure and pesticide use in these fields. And we found that different times of year and at different levels, we very frequently did find that pollen in these areas was contaminated with a few different pesticides, unsurprisingly, given that we know these pesticides are used. One is chlorpyrifos, an organophosphate insecticide that was briefly banned in the United States, but then came back, and so it may or may not be here for the long haul. Thiamethoxam, a neonicotinoid that's used primarily in seed treatments, and lambda-cyhalothrin, a pyrethroid insecticide that's really commonly used in foliar applications to treat a variety of different pests. At the same time, we also saw a lot of azoxystrobin and pyraclostrobin, two fungicides frequently, again, used in both of these crops. And so Ge Zhang and Mara Hall did a lot of this work and showed us what those levels might be. And so now looking at this system, we could kind of start to put things together and create a laboratory facsimile of what might happen in a real world setting, where you would have honeybees, again, using them as our model pollinator, that are exposed to variable flower resources, whether prairie strips are present or absent. They're exposed to variable levels of pesticides, and we know they're ubiquitously compared to parasites, or exposed to parasites and pathogens. And so a PhD student in my lab named Ed Shea did this really nice series of very annoying and complicated experiments to combine these three factors together and see how they interacted. And this work basically resulted in us using two model diets, either real pollen or a protein supplement like Mega Bee, using different pesticide combinations, chlorpyrifos or fungicides, these two different insecticides, and then exposing bees to IAPV infection, yes or no. And this paper just came out in the Science of the Total Environment. And I don't have time to talk about all of it today because Ed did lots and lots of experiments. But some of the take-homes are as follows. So first off, if you look at bees who are under what we consider to be probably nutritional stress, just feeding them a protein supplement, no real pollen, we see this kind of apparent synergism where if bees are given, no virus or either with or without a pesticide, there's no real effect. So I highlight this, that the pesticide levels that we're giving these bees is not so high as to cause mortality. It really doesn't. These are truly sublethal pesticide levels. However, when you introduce the second stressor of a virus infection, again, controls, a lot of them do die because IAPV is deadly to them. However, if they've also been exposed to these sublethal pesticide levels, they have a significantly higher amount of mortality. So we see this kind of synergism or additive effect of pesticide stress and virus infection stress. However, if we look at this with bees who are fed what we basically consider the highest quality pollen we can get our hands on, not only does that effect go away, it actually reverses. So bees who are on a high quality diet, if you give them a pesticide treatment, it makes them survive the virus infection better. So we see that in here. It's not a huge effect, but it is significant. And this is not totally unbelievable. There's a lot of literature in the pest world and especially in the heat stress world about this phenomenon called hormesis, where a small amount of stress from one factor that's not lethal helps an animal prime its stress response system to become more resilient to later stresses. And so what we think is going on here is that a low dose of a pesticide can prime a bee for other stresses, but they need the right nutritional environment to actually be able to have the resources to do this. Interestingly, though, it doesn't replicate across different pesticide classes. So chlorpyrifos, lambda-thiolothirin, thiamethoxam, these are all different insecticides with different modes of action or from different classes. And we see that the effect is not the same across them. I highlight this because I don't want the conclusion to be that, yeah, a low dose of pesticides is actually good for bees. It's more to highlight that it's actually much more complex than we know, that the sublethal effects of these different pesticides can be very different. And I know I only have a couple more minutes to wrap up. The other thing I want to highlight, though, is that we can ask, well, what is honeybee health? That's a very contrived lab study, right? Just testing this bioassay of how they respond to a virus infection. But there's a lot more to what makes especially a honeybee colony successful. And one of the big ones I want to highlight that we really don't have very much resolution on is reproduction and how the queen and her health is affected by all these different factors. And I had an amazing postdoc named Ashley St. Clair who was in my lab and did some really awesome work on this. And like I said, I don't have tons of time to talk about it right now, so I'm going to skip ahead. But we we did some really nice work utilizing these tools called queen monitoring cages that were developed by Gene Robinson's lab, and we did a lot collaborating with Julia Fine, who's now at USDA ARS, in using these basically highly specialized microcolonies that allow a queen to lay her eggs, and that we can assess egg laying, to test how the same factors that I just talked about affect queen health. And basically, I'm going to skip ahead a little bit for time's sake. We found that that same pesticide mix that caused, I'm going to call them benefits to workers, caused significant declines in queen egg laying and caused a significant increase in failed hatch rate, so developmental deformities in her larvae. And so, while we might see that a pesticide exposure event maybe either has no effect or even improves response to virus infection, it can have a totally opposite effect on reproductive rate inside that same colony. Again, showing that there's a lot of different moving parts in identifying what phenotypes are most important or how they're affected by these different things. The other thing I just want to very much highlight, again, running out of time, my Zoom errors, is how important these pesticide questions are to queen bees, because of how we view queens from a regulatory and policy perspective. And I highlight this because I think this is of interest to the group, because I saw the mention of policy in it. So if you were to go into the United States Environmental Protection Agency's guidelines for how we assess pesticide risks to bees, you will find this cool chart. And it tells you for a larva and for an adult of worker, drones, and queens, how long they're in different stages and how much food they consume of royal jelly, nectar, and pollen. And if you look in this, based on some real literature, queens are assumed to consume no nectar and no pollen ever as adults. They are expected only to consume royal jelly. However, we did a series of experiments in the lab and in the field showing that, believe it or not, queens consume a significant amount of pollen. We did this in this comically simple way of just feeding bees pollen with food coloring in it. And Apis mellifera ligustica is transparent enough that you can just hold them up to the light and see how colorful their abdomen is. And so we can see that within 1 to 2 days, pollen and nectar are being very easily observed inside their guts. We then went, and I'll skip ahead for this, and sacrificed about 50 field queens from multiple different parts of the United States, dissected them, and then stained their guts for pollen. And we compared that to workers from the same colony. Unsurprisingly, workers have way more pollen in their guts, but queens, either in the field or in these queen cages, also have quite a lot of pollen in their guts, kind of highlighting that how have we been kind of underestimating what a queen eats for so long? It's kind of shocking to me. If you look in the literature, there's very poor resolution on what does a queen eat. And this is, again, still kind of shocks me. How did we not have a definitive answer at this point for what the queen honeybees eat? We don't know. But it has these really important regulatory implications because it's assumed that queens don't eat any pollen, they don't eat any nectar. Therefore, their exposure to pollen with pesticides in it is zero. They have no exposure, therefore no risk. And so we're seeing like that's very unlikely to be true. So again, I want to wrap it up so we still have some time for questions. Again, I've had a lot of collaborators and a lot of great lab members to work on this. We've had good support from a variety of different agencies. And it's been, again, it's been a lot of fun to get to answer these questions and start to look to the future of how we can better understand bee pathology and bee response to pesticides in these real world environments. All right, I'm gonna stop talking.
Excellent. Thank you so much, Professor Dolezal. If you could all join me in a big round of applause for Adam. Yay. What a very informative and comprehensive talk. Okay, we have some questions, and I'll remind the audience that if you do have a question, please go to the bottom. There is a Q&A button. So if you could type in your question there. We already have a couple of questions. The first is from Dr. Clement Kent. He asks, do mites feeding on IAPV-infected pupa emerge successfully if the pupa never mature?
That is a great question, and I think, and we don't actually know. So this is kind of why I'm really interested in is, and I didn't again have time to talk about this, but this benefit of manipulating host behavior and host development, it probably benefits IAPV, but we know that IAPV is primarily being transmitted by Varroa, and so it also makes sense that they may play some role if there's some coevolution going on, because if it can maximize the time that a bee is a pupa and increase the developmental for Varroa, that's going to be very beneficial to Varroa. The big question we have now, and we're trying to test right now, is if a bee exhibits this developmental arrest, she's under a cap in the real world. There's a cap under her. Do the workers in the colony recognize her as sick and try to use hygienic behavior to remove her? Or are they able to manipulate how that larva smells or appears so that they remain unmolested by the workers? Because if that's the case, I think it would really maximize the replication time for IAPV and have this potential effect on Varroa. We actually don't know, but I think this is a really interesting question.
Excellent, thank you. I think we have another question from Dova, and I do see that somebody had, I think Wanja, you had put your hand up. If you're able to type the question in the Q&A box on the bottom of the Zoom platform, that would be much appreciated. Thank you. We have a question from Dova. Do you know of any studies, or are you studying the genetic differences between the bees that survive the viral infection and the ones that don't?
So yeah, we don't work on this. I believe that Ishmael Amiri at Mississippi State is now working on this specifically with IAPV. He came from one of the other labs that did a lot of IAPV work. I think he's really interested in studying. I think they have like a USDA project they just started, trying to, I think, look for genetic lines that may have greater resistance to virus infection. There's been studies with the deformed wing virus more than IAPV, both artificially selected but natural selection based. And in the DWV world, I think what they've often found is there may be genetic lines that are, I want to use the in finger quotes, resistant, but in most cases, they appear to be tolerant of very high deformed wing virus loads, which is not really ideal from a bee breeding perspective. And so those lines do exist, but I think there are some challenges to, I don't know, like using them practically. Anecdotally, there are huge colony effects. If you can use day olds from two different colonies, and their response to infection will be very different. We also had a project that I didn't have time to talk about where we measured the response to infection in the same colonies every month for 14 months. And the same colonies with the same queens, their bees will respond very differently across time. So disentangling the genetics may be actually hard because the environmental effects are so huge. Yeah.
Excellent. Thank you, Adam. We have another question from Aiden. What could be the mechanism behind guards letting infected bees into the colony? Is it pheromones versus behavior versus potentially others?
Yeah, this is a great question. When we first saw this phenomenon, we at first thought maybe it was a behavioral difference in the infected bees, but I don't think it is. What we see in the infected bees is that it changes their cuticular hydrocarbon profile. So we know that when bees are immunostimulated, people have done this for a while, inject like bacterial coat proteins into a bee, their hydrocarbon profile changes. So now they smell different. It usually elicits greater aggression because they smell sick. That's bad. When we infect bees with IAPV, we also see a shift in their hydrocarbon profiles. Hydrocarbon profiles are complicated, so we don't know if it's not like one magic compound, but it does change some compounds like octocosane that have been associated with greater acceptance by guard bees. And so what we think is that the virus infection is changing their hydrocarbon profile to make them smell more neutral, if you will, and therefore get accepted more by these other guards. And again, this is like a little bit more preliminary data, but we were trying to figure out the mechanism by which they could actually change the hydrocarbon profiles. And in the interim, from when we started this project to finishing it, we originally were going to do some like bee transcriptomes and look at bee gene expression. But in that time period, Cassie Vernier from Yuda Ben Shahar's lab did this really cool set of studies showing that the establishment of the gut microbiome during the newly emerged phase really shapes the hydrocarbon profile of bees. And so that really is a big part of determining their colony odor. And so we did some experiments seeing if the microbiome was disrupted by IAPV, and it is. So when you infect a bee with IAPV, their microbiome is also changed, and that appears to maybe have effects on this hydrocarbon profile. So we're starting to get some like little dots along the line to figure out kind of the mechanism from virus to hydrocarbon. And you asked, is the queen egg laying and pesticides published yet? No, it is not published yet. It should, yeah, for better or for worse, Ashley got a permanent position job, and so she left to go do that job, and so we've been a little slow in getting that work out, but we're hoping to submit it pretty soon.
Okay, we have another question. You mentioned the impact of native habitats on mitigating colony declines. Could you discuss the implications of prairie versus agriculture sites on colony health, and how habitat restoration might be integrated into conservation practices? This is from AK and Nixon.
Okay. Yeah, I think that's a great question. So again, we saw pretty big effects in honeybees when they have access to these prairie areas. And I didn't show this paper, but there was a paper by Gu Zhang. His advisor was Matt O'Neill, who's the picture of right here. And he showed that when you have bees and honeybees kept in these areas with prairie strips, you can also see significant increases in honey yield, again, suggesting that, just overall health is better. I think that it's probably going to be highly variable in the agricultural landscape that you're in. So the area in like Illinois and Iowa is interesting because, as I said, it's extremely extensive. There's a huge amount of land dedicated to corn and soybean production. However, believe it or not, our insecticide use is actually pretty low in comparison to other parts of the world that grow these crops, because of resistant genetics and BT and things like BT corn. We actually don't apply that many insecticides. If you were to go to like the southeastern US or to Brazil, insecticide applications are much more frequent, and the pest pressures are very different. And so you may actually see a very different effect there. So I think that that's something I've had to really try to stress, is that these habitat restoration recommendations are going to require local research. You're going to have to look at your area and what's successful there.
And we have another question from Wanja. What can conservationists do to safeguard pollinators in agricultural areas?
Yeah, so this has become, very controversial. Again, in our strips work, there was even talk of recommending against putting in prairie strips because of this concern about ecological traps, that you'll grow these great flowers, that'll attract bees, and then they'll be damaged by insecticides. We don't see a lot of evidence of that, but I think it is really important that if somebody's going to put in agriculture or conservation land into like adjacent to an agricultural area, that they'd be thinking about doing those best practices. And for example, in all of our sites, they're primarily owned by farmers. A farmer has put in this prairie strip, it's theirs, they own it, right? And so those people are doing things like applying insecticides per all the best practices so there isn't spray drift into that area. But that's not always the case. And so I think it's going to be like really making sure that there's buy-in from the people in the land next to you, that they are going to actually abide by those practices and kind of appreciate that.
Excellent. Thank you so much. Please join me in a round of applause for Adam. That was a fantastic presentation again. Thank you so much for sharing all your knowledge and about viruses and pesticides.
Yeah, thanks everybody. Sorry I got a little disjointed. I got a little flummoxed with the Zoom thing, but hopefully I didn't run too over.
No worries. Thank you so much.
Bye guys. Thanks again.
So once again, Adam, thank you very much for such a wonderful and informative presentation. And I just wanted to remind everyone that next month we also have another speaker series presentation. This one is by Dr. Laura Figueroa on Friday, October 11th at 11 a.m. Eastern. She is an assistant professor at the University of Massachusetts Amherst and will be talking to us about the fascinating world of bees, from novel monitoring tools to landscape level stressors. In order to register for this webinar, please visit our website. That's yorku.ca/bees. And just a week afterwards, we have our big yearly conference, BeeCon. So as a reminder, BeeCon is a hybrid event with a virtual day on Thursday, October 17th, and then an in-person at York University or virtual day on Friday, October 18th. Our keynote speaker this year is Dr. Chelsea Cook, who is an assistant professor at Marquette University, who will be discussing social animals and if they are able to manage a changing environment more effectively than non-social animals in the context of bees, as well as 41 great additional talks centering around all things bee. In order to register for this, please visit our website by September 30th, 2024 to be able to attend virtually or in person on one or both of these days. And with that, I would like to thank everyone for joining us today. And we hope to see you again next month for our next speaker series and BeeCon. Thank you.